So it's time to put together some of the ideas we've learned in previous lessons to map out the formation of a human memory. Here's a human brain over here, and it's just amazing to think that this tissue can store lots of what we call memories. We can learn things about the world. There are changes that happen in the brain, and then we can retrieve information that was stored from a learning event. We'll call this the anatomy of memory. We'll look at 3 stages of the process: the encoding stage, the storage stage, which will involve plasticity between neurons, retrieving a memory, and then a very important process called system consolidation that will strengthen memories in our cerebral cortex. We've also learned about a couple of famous neurological patients, H.M. and Clive, and they both had a similar kind of problem. Because of the damage to the brain, the hippocampus and surrounding tissue, they have a failure to be able to write new information to their hard drives. The hard drive would be the cerebral cortex here, and they can't sort of make any new episodic memories. They can't store new information to the hard drive. They can access stored information from their semantic memory, so they can carry on conversations and such, but they can't write new data to the hard drive. In the brain, then, the problem is both of them suffered damage to the hippocampus and the surrounding temporal cortex, and again, so conceptually, they can't add anything new to the long-term memory system in the cerebral cortex, what we're calling the semantic memory system. We've also learned a little bit about the short-term memory system that we called working memory, and we're going to use in this lesson a typical sort of classroom experience as our example, where we're sitting down at a microscope. We might be looking through the eyepiece and we see single-cell creatures interacting. The amoeba is going to be eating the paramecium, and maybe the teacher is saying something while we're observing, naming these creatures amoeba, paramecium, and the amoeba uses pseudopods, these extensions of the amoeba's membrane, to capture its prey. So we've got a learning event, a learning episode here. We've learned that the visual cortex and auditory cortex and the touch cortex are going to be sending results of their processing up to the working memory system, and will have a conscious experience of the event. So whatever is in the working memory system has the potential to be stored as a long-term memory, and that's because the areas that are processing the information and sharing with the working memory system are those same areas that are going to be storing memories for the event. So we're going to end up with, as a result of this experience, memories, what it felt like to handle the microscope, memories of the sounds of the words that were uttered, visual memories of what we were seeing under the microscope. So now we're ready to map out, to trace the formation of a memory of this learning event. In this brain, then, we have in the frontal cortex here the working memory system, and then I've drawn these circles here will represent populations of neurons in the touch cortex, visual cortex, auditory cortex, and here we have our hippocampus with hippocampal neurons. So the first stage, then, is going to be the encoding stage. A subset of cortical neurons respond to the learning episode, and some will be allocated to a memory trace. So here we have a population of neurons in the touch, visual, and auditory cortex. Notice some of them are colored in. They're showing to be activated by the learning scene. So some of the visual cortex neurons are being activated by the site of the amoeba and the paramecium. Some of the touch cortex neurons are activated by the handling the microscope. Some of the auditory cortex neurons are activated by hearing the word amoeba and pseudopod, etc. So the idea then is that out of a population of neurons that are processing the various sensory qualities of the experience, a subset of them are going to be allocated to forming a memory. And notice the memories will be sensory specific. The site of the amoeba and paramecium, the memory for that will be stored in visual areas, the touch of the microscope in the touch areas, and the sounds of the words in the auditory cortex. The word allocation then is just referring to the fact that there's going to be a distribution of responsibilities among the population here. So in other words, only some of the neurons are going to have the job of forming the memory trace. And so that's what it means to be allocated, that some kind of responsibility is being assigned to some members of the population and not others. In previous lessons, you saw this video of engineered neurons. So here we have a bit of the brain on the left here. Here's a magnified version of that. I'll play the video. These neurons had been engineered to emit light when they generate electrical signals. So we can kind of visualize the firing, the electrical activity of populations of neurons. The big structures here would be blood vessels carrying blood. So the little flashing lights are brain cells. So we want to imagine this population of neurons could be in the visual cortex, could be the touch cortex, could be the auditory cortex. So when we see something, a certain population of neurons responds. Not all of the neurons in visual cortex, just a subset of neurons respond. And out of those neurons that respond, a subset of those neurons will be allocated to form a memory of what it was that we saw. Now, the activated neurons in each of the cortices here, not only are they going to be sending information to the working memory system, so we're having a conscious experience of what's happening, but they also send information down into the hippocampus. So notice then, the activated neurons in visual cortex are going to share their activity with the hippocampus. Touch cortical neurons sharing their activity with the hippocampus. Auditory cortex neurons sharing their activity with the hippocampus. So we have a convergence. So the allocated neurons communicate their activity to the hippocampus. The exact pattern of activity represents what the learning event sounded like, looked like, and felt like. The hippocampus therefore receives all the information that will be linked to form a memory of the event. Notice then that the hippocampus is situated so that it can link together the different sensory aspects of the event into sort of like a unified learning episode. A brain without a hippocampus then cannot link sensory information into a unified memory, and HM and Clive therefore could not make new memories. So you see, if your hippocampus is damaged, then you've lost the ability to be able to make a memory that kind of connects up the different sensory qualities that your cortices are processing. Now, scientists suspect that the most excitable hippocampal neurons at the time of the learning are the ones most likely to be activated and form a memory of the unique event. So we have another allocation process going on here, just as we saw in the cortex. And allocation, we can think of that as just some brain cells, in this case hippocampal neurons, are going to be assigned to a certain task, and that task will be to make a memory of the particular event. Now, the activated hippocampal neurons undergo a growth process that strengthens their connections to each other. And hippocampal neurons are very good at responding to a single learning event. And neuroplasticity is the basis of learning and memory. So up here in our picture, before learning, this would be a typical synapse between two hippocampal neurons. After learning, as a result of the learning episode, there's a growth. That hippocampal neurons that were activated by the learning event enjoy a stronger connection as a result of that growth. And they're very good at growing a lot with one learning event. So we're identifying that as high plasticity in the hippocampus. We are now entering the storage phase here. So we were encoding information, so certain sets of neurons are responding and analyzing the scene and then activating the hippocampus. Now with neuroplasticity, we are beginning the process of storing the memory. As hippocampal synapses get stronger, they are forming a memory trace of the episode. So again, here we have a couple of hippocampal neurons. Notice the green one here was getting auditory information. The blue one here was activated by touch sensory cortex information. The red one here was activated by visual cortex information. And so the hippocampus has a sort of a network architecture, and we'll imagine these hippocampal neurons are connected to each other in ways that can be strengthened as a result of the learning event. So the hippocampal neurons that were activated get stronger connections with each other. They grow stronger synapses. So after learning, the network of hippocampal neurons is more strongly connected. That is the formation of a memory trace. So we're left with a strong memory trace in the hippocampus indicated by solid lines between the neurons that were allocated in the hippocampus to store the memory trace. But what happened in the cortex? Well, notice I've drawn some sort of dashed lines between these neurons for a reason. The cortical memory traces are still weak at this initial time. So there was less plasticity in the cortex compared to the hippocampus. Again, the hippocampus has an interesting quality. The hippocampal neurons are really good at growing a lot in response to learning events, so they can make strong synapses, forming strong memory traces with just one learning event. If we were to peer into the synapses up in the cortex in the different areas here, take the visual area, before learning there would be some baseline strength of synapse. After learning there is an increase, but it's a small increase in the strength of the synapse, shown by slightly larger terminal there. So we've got some plasticity in the cortex, but much greater plasticity in the hippocampus. Now the hippocampal memory, HC for hippocampus, the hippocampal memory trace is like an index that is connected to the cortical memory traces of the event. So the index represents the pattern of cortical activity that occurred during the learning episode. So it's kind of like a way to kind of keep track of what the cortex was doing during that learning event. So this strong hippocampal memory trace is connected to the cortical ones, and that will become important, as we'll see. Because cortical memory traces are weak at this time, the hippocampal memory trace is required for memory retrieval. So we'll see later that there are other brain systems that can access cortical memory traces, but in this early stage, this hippocampal memory trace is going to be needed in order to access the cortical memory traces. Damage to the hippocampus at this stage would likely make the cortical memory traces inaccessible, and this explains why HM and Clive had amnesia for events just prior to surgery, or for Clive just prior to his viral infection. The cortical memory traces were weak. So you see, if at this early stage you damage the hippocampus, you've eliminated the index. And because these cortical memory traces are weak, other brain systems can't access them, can't reactivate them. So you will show amnesia for things you learned just prior to hippocampal damage. After learning, so now let's take a look at what happens when we're retrieving a memory now. So we've completed the storage phase. What about retrieval? After learning, a retrieval cue, in this case maybe the sound of the word amoeba, activates one part of the cortical memory trace. So here we see the sound of the word. Maybe it's the next day in class. The teacher says, what do you know about an amoeba, right? Or think about the amoeba you saw yesterday. So the word amoeba is going to activate auditory cortex. There will have been a memory trace of the sound of the word amoeba, so we're going to recognize that word. We heard it yesterday. Activity in the auditory cortex neurons will be sent down to the hippocampus, and notice it's the green neuron here in the hippocampus that will be activated. Right, so one of the hippocampal neurons in that memory trace will be activated, and then because hippocampal neurons in the memory trace are strongly linked, activation of one neuron reactivates the entire memory trace. Scientists call this pattern completion. So the activated index then can be used to reactivate the cortical memory traces, as we'll see in a moment. But this is critical. Notice the cue was just activity in the auditory cortex, the sound of the word amoeba. But if it activates one of the hippocampal neurons, because of strong connections in this network down here, the entire memory trace can be reactivated. And you might kind of guess what's coming next. Well, remember the blue hippocampal neuron is connected up to the cortical memory trace, and the red one here, that's in the touch cortex, and the red one is connected to the cortical memory trace and visual cortex. So the activation of the hippocampal memory trace is going to be used to reactivate the cortical memory traces. And we'll draw that like this. So during retrieval, information flows from the hippocampus back to the cortex to activate cortical memory traces. So there's a two-way communication between the hippocampus and the cortex. The reverse information flow occurred during encoding. So notice during retrieval, we're using the hippocampal memory trace to activate the cortical memory traces. But let's remember what happened during encoding. It was the reverse flow of information. Cortical neurons were activating hippocampal neurons. So when we reactivate the cortical memory traces, we'll say that the memory is being reconstructed. Notice then, activated cortical memory traces will send activity to the working memory system, and now we will we'll have the conscious experience of remembering the learning event the day before. So just hearing the word amoeba, it was just auditory cortex that reactivated the hippocampal memory trace, but that successfully activated all the other cortical memory traces. And now I can remember sitting at the microscope and looking and imagining and kind of reliving what the amoeba and the paramecia looked like, and I can recall hearing things about these organisms, so I can retrieve an episodic memory in this case if I'm remembering the circumstances of learning. Now sometimes retrieval fails. So after only one learning event, retrieval might fail if cortical memory traces are not successfully reactivated. So one way to think about failure is just this communication was not successful between the hippocampus and the cerebral cortex. But there's another way failure might happen. The hippocampal memory trace might also be weak if the student was not paying attention during the learning episode. So the quality of the hippocampal memory traces is influenced by several factors. So attention is one of them. So if we were sitting in class, yes, the microscope might be in front of us, but we weren't really, paying attention to what was being said, or we didn't really look carefully, we might have not established a strong hippocampal memory trace in the first place. And if we don't have a good, strong index here, we're not going to be able to successfully reactivate cortical memory traces. Other factors can influence the quality here as well. Alcohol, for example, interferes with the plasticity going on in the hippocampus, and so alcohol has amnestic effects. High levels of alcohol will make people forget what happened to them because the hippocampal memory traces are not forming very well. So for reliable memory retrieval over time, cortical memory traces must be strengthened by a process called system consolidation. The system term captures the fact that a memory is being reorganized across multiple brain areas, in this case hippocampus and cerebral cortex. Consolidation refers to the strengthening of the memory traces when the synapses get stronger. So system consolidation is the key then to making long-term memories in the cortex. It's the process that's going to strengthen these initially weak cortical memory traces. The hippocampus, again, is going to be a key player in this process. So we'll look at system consolidation in the next part.